Welcome to Lecture 2. In this lecture we'll cover numbers in biology, scientific notation. We'll talk about biodiversity and classification in biology. So numbers in biology. So biology has three types of numbers, so it's three groups of numbers, that two of which go into the extremes and one of which is a normal number. For example, let's say you are a behavioral scientist and you are looking at you examine the gorillas in the mountains in Central Africa, and you have a gorilla group of 20 gorillas. This is a fairly normal number, and so if you write your report, you write 19 gorillas or 20 gorillas, and 5 gorillas went off and 6 gorillas came back. So these are fairly normal numbers, they are easy to write. If you're a molecular biologist and you're writing about the enzymatic processes, the enzymatic functions, metabolic functions within a cell, there are millions of those within a minute. And so it is very hard to write those numbers. If you write millions, you don't always want to write 1, 0, 0, 0, 0, 0. And the same is true if you are working with very small numbers. For example, you are looking at the structure of a cell under the microscope, and there is a little organelle that is something like 50 micrometers in diameter. You don't want to write, okay, this is 1/50 millionth of a micrometer. You have to have some way of expressing these numbers. And this is scientific notation that helps us with this. So the concept of scientific notation, and you've certainly learned this in high school, is you can express 100 or 1,000 as 100 or 1,000 and write the zeros down, or you can express this as 10 to the power of 2 or 10 times 10, or 1,000 10 to the power of 3 or 10 times 10 times 10. And you can do the same for higher numbers. For example, if you are talking about the number of neurons in an organism, in a human, there are an estimated 100 billion neurons. And you can write this down in this way, in a long way, or you can take scientific notation and say, okay, I have 1 times 10 to the power of 11 neurons within a human organism. And we tend to do this rather than writing all of these zeros. Same thing with the numbers of cells in a human body. An estimated 60 trillion cells on average in a human organism. You can write this in this way, or you can take scientific notation and you can write it in 6 times 10 to the power of 13 instead. So this helps us to express large or small numbers. And small numbers, as I said previously, if you look at under a microscope and you look at structures such as those neurons here, they are usually about in the micrometer range. This is the light microscopic resolution that you will see. You see millimeters or micrometers. And it is important to note that in the sciences, in the natural sciences, the universally adapted measurements are metric measurements. So we will not measure in inches or yards or feet. We'll measure in millimeters, micrometers, nanometers, centimeters, meters, kilometers. So this is the metric system. And micrometer is 1 millionth of a meter, or expressed in scientific notation, 1 times 10 to the power of -6. So to give you an aid, if you are at a microscope, a dissecting microscope, a light microscope, usually the structures that you see are either millimeters under the dissecting microscope or micrometers under the light microscope. There are even smaller structures, and as I said, it's organelles, for example, or little structures within cells that are much smaller than that, and they are about 1 billionth of a meter. And these would be nanometers, 1 times 10 to the power of -9. And so when do we work with nanometers? When we look at electron microscopic structures. So in this case here we see a synapse, and the synapse we see a mitochondrion here, the cross-section of the mitochondrion. And here we see individual synaptic vesicles. And these would be about 20 or 30 nanometers in diameter. So if we look at an electron micrograph, we usually look at nanometers or a billionth of a meter. If we look at light microscopic structures, we are looking at micrometers or millionth of a meter. And in order to make that easier for you to familiarize yourself with all those measurements and this nomenclature, I have this table here for you where you have here you have your pico, nano, micro, milli, centi, decimeter, meter. And here you have the numbers of the zeros behind the 1 in order to express this number, or the number of zeros after the punctuation here in order to express those numbers. Here's the scientific notation, and here it tells you, for example, the micrometers here is a millionth of a meter, the nanometer is a billionth of a meter. So if you are in any doubt, you can pull up that table on your Canvas site and you can look at it. I've highlighted here in red, or boxed in here in red, the numbers in which we usually work in the biological sciences. And when we work with a microscope, we usually have micrometers. So let's move on to biodiversity. There's an estimated 3 to 100 million different types of animal species that inhabit our planet. Scientists don't really know how many there are because all the habitats are not explored yet, and particularly the smaller animals, the microorganisms. Scientists literally on a daily basis identify new species, particularly in the rainforests or in the less well-explored deeper sea habitats, where a surprising amount of different species is being discovered. About 400,000 different plant species have so far been discovered, and interestingly, the life can be subdivided into eukaryotic organisms and prokaryotic organisms. And eukaryotic organisms are the higher developed organisms, and they are different from prokaryotic organisms insofar as they have organelles within them that are surrounded by cell membranes. Whereas prokaryotic organisms in general don't have those. There are exceptions, but in general they don't have organelles within inside of the cell that are surrounded by membranes. The eukaryotic organisms can be subdivided into the plants, the fungi, animals, and single-celled organisms, the protists. And the prokaryotes can be subdivided in the archaea, or you will also hear these are extremophiles. They live in extreme conditions like in hot vents. And bacteria obviously are present everywhere. They are good bacteria, there are harmful bacteria. So we'll talk about bacteria when we talk about prokaryotic organisms. The biodiversity that has emerged over time was acknowledged by early explorers, particularly British explorers, and one of the key figures in postulation of the hypothesis how this biodiversity has been created on planet Earth was Charles Darwin. And Charles Darwin has conducted extensive studies throughout his lifetime, really, and has written a book that is titled The Origin of Species by Means of Natural Selection. And within this book, he postulates his hypothesis how different species have emerged over time. So what he postulates is that organisms change over time, and the best adapted organisms survive and pass their genetic information on to the following generations. Charles Darwin lived from 1809 to 1882, and his most crucial findings that he has summarized in his book, The Origin of Species by Means of Natural Selection, he has collected on the Galapagos Island, that was part of his 5-year expedition on board the HMS Beagle from December 1831 to October 1836. And he himself wrote that the voyage on the Beagle has been the most important event of my life and has determined my whole career. And it is important to acknowledge that during this time where Charles Darwin was active, the belief in the creator theory was prevalent, and in parts of this world still is prevalent, so that there's one, the church believes in the creator, and then Charles Darwin came up with this theory. Well, I believe that species have evolved through advantages that they had during development. And here's a classic example that he brought. On the Galapagos Island, he has observed that some islands have tortoises with very long necks, whereas other islands have tortoises with very short legs. And what he has observed is that the tortoises, the islands that have the tortoises with long necks are drier and the vegetation is more sparse. And so his conclusion is tortoises that developed or were born with an abnormally long neck had an advantage on those dry islands because they could utilize the natural resources that were located, or the food resources that were located higher on the plant, because they could reach them and had an advantage over the shorter neck tortoises. For that reason, this advantage made them reproduce more successfully, whereas the shorter necked tortoises could not compete with those longer necked tortoises and got extinct on these islands. On other islands where that are not that dry and where vegetation and food resources are not such a problem, the shorter necked tortoises have still survived. And it was those observations that he made and his conclusions that he drew from these observations that led him to his conclusion that there's natural selection, and this natural selection leads to differences in organisms, or to the expression of, or the determination of certain traits within those species that make them survive better. And he hesitated to publish this because of this prevalent creator theory that at the time that the church had. And it was another British scientist, and his name was Russell Wallace, or Alfred Russel Wallace. He was also a British naturalist, and he lived from 1823 to 1913. And his studies, very similar to those of Darwin, were mainly conducted here in Southeast Asia. And he came to similar conclusions. And what he did is he wrote this letter to Charles Darwin and summarized his conclusions and brought his idea to Darwin. And when Darwin saw his letter, and he had already written parts of his book, The Origin of Species, had just not dared to publish this and come forward with this, then prompted by this letter from Alfred Russel Wallace, he then, both of them, presented their findings at a conference, and after that Charles Darwin published his book on the origin of species. So biodiversity, there's a very large biodiversity on this planet, as I mentioned earlier, and obviously this biodiversity, scientists have put an effort in categorizing this biodiversity. So this is the classical tree of life, where you have the protists that are at the very basis of this life of tree. And then there are certain branches that branch off. There's the animals, and there are the plants, then there are the single-celled animals, there are the sponges, there are the jellyfish and the cnidarians, and then there are the mollusks and the insects, the arthropods, insects, chelicerates, and then we have the mammals. So they all have to be categorized in a certain way. And it was, for the longest time, cladistics that was used in order to categorize these. Nowadays, these trees look very different because now that genetic methodologies are available, we are now able to use genetic information in order to identify which animals are closely related to which animals, or which organisms, living organisms, are closely related to which organisms. So cladistics was used in order to get these general phylogenetic trees. And I'll give you an example here. So how do you classify an organism? And this is the example of the eastern gray squirrel. So here you have your tree of life. And first of all, you determine, is it an animal or is it a plant? So here, first of all, is it a eukaryotic or prokaryotic organism? So here, this gray squirrel belongs to the eukaryotic organisms. Then you look, is it an animal or is it a plant? So it belongs to the animals. You now go higher up here in the tree, and you now look at the phylum. And this now does not belong to the invertebrates, it now belongs to the chordates. They have a vertebra. These are mammals that have mammary glands and fur. They are rodents. And then you go further and further down into your species until you're at the very tip of your tree of life, and can then determine what kind of specific species it is. And the way how these trees were formed is through cladistics. And what you can see here is a so-called cladogram. And this cladogram now categorizes your animals and allows you to see which ones are more closely related to each other. So here, for example, let's say all of those have a backbone. So, but if you look at this hagfish here, what this hagfish has different from, in which way it differs from all the other animals, is that it doesn't have a jaw. So for that reason, it has its own clade, and it is separated from all the other animals. So all the other animals are carried forward. Now we look at the next animal and we say, okay, what is different here from all the other animals? And this is that this animal has gills, whereas all the other animals have lungs. So for that reason, fishes are now a different clade. Then we have this animal here that does not have claws or nails, the salamander, compared to all of the other animals. So for that reason, you now branch off the salamanders here. What do the next ones have now in common? They either have feathers, fur, or mammary glands, whereas the lizards don't. They have scales. So in this way, biologists have now categorized life on Earth. And this concludes our second lecture, and I will see you in the third lecture.